Good morning, everyone. I'd like to thank the organizer and I guess especially Nagla for giving me this nice title that you see. Is it time to move on from HCV? And I guess for anyone that think about these questions, the first thing that came into your mind is, well, when you invest a lot of time in anything, it's difficult, I guess, to move on something. And I'd like to start with this first slide, which is a figure that I've just prepared for a review articles on HCV protease inhibitor for the 25th anniversary of the discovery of HCV through an invitation from Stan Lemon and Chris Walker. And it roughly describes the timeline to these, from the disease to these major breakthrough, which we call all oral therapy. And you can see on that slide that represents almost 40 years. And I'm pretty sure there's people here in the room that were there from the beginning, talking about this non-A, non-B disease, and up to last year, 2015, is where we do have these breakthrough therapy. So personally, I started working on HCV at Boehringer Ingelheim in 1995. So that's 20 years ago. And so my major contributions for the last 20 years have really been in HCV. So how can we, I guess, recycle all this knowledge? And this is a problem for many people. So in our lab, our main goal is the discovery of broad antiviral compounds. And I would say 20 years ago or even 10 years ago, that's mission impossible, very difficult. Now I think we do have the tool for this challenge. And in the lab, we have two major research programs. One which is based on trying to identify novel regulator of innate immunity, which I won't be talking today. And the other one is really based on virus-host interactions, which, as you know, viruses are totally dependent on cells. And in cells, most of the process are taking place through protein-protein interactions. So by, I guess, studying virus-host protein-protein interaction, you get a good overview of all the process that the virus can either induce, block, hijack in order to survive. And he does it very well because it's the result of a co-evolutions in between the host and the virus, such as almost like a pharmacological tool. So in the last, I would say for the last 5 or 10 years in our lab, we've mainly been focusing on trying to better define these virus-host protein-protein interactions, and we use HCV as a model, as a virus model. And this is one of the last articles that we published 2 years ago now, which is elucidating the HCV, we call it the HCV interactome. And indeed, it was a study where we've looked at host proteins using all HCV protein that has their ectodomain in the cytoplasm of the cells in order to identify some novel host interactor. And we did it with state-of-the-art mass spectrometry techniques with Pierre Thibault at IRIC. And like many of these studies, identified a few hundreds using different criteria. But what you have on the slide is a set of what we call very specific proteins because we did it for, like I said, for almost 6, 7 protein, all the non-structural proteins. And so for these type of data where you can generate a lot of false positive, each protein controls for itself. So if you have a host protein that interacts with every viral proteins, you deprioritize this protein. So what you see here, it's kind of a heat map of roughly 100 proteins, and for which the color indicates really the interactions with the name of the proteins on the right, but it doesn't really matter for this talk. But you realize that there's a good number of proteins from the core, for example, which are interacting with host proteins, and the same thing for all the others, and they're all quite specific for these viral proteins. We were in collaboration with a group, an excellent group in bioinformatics at Lyon, the group of Vincent Lotto, which they have a whole database of all virus-host interactions, especially also for HCV. We've saw that for these hundreds, there was 24, or let's say 25%, which is a pretty good rate of host protein that were already reported in the literature. So showing that this approach was quite robust. And if you remember in the HIV fields when they were using these type of system biology approach, RNAi screen, for example, and published in Nature Cells, they end up with no overlap at all in between all these approaches. So having 25% is among a very good overlap with the literature, indeed. And there's still 74, which are novel protein that has been discovered. So in collaboration with that group, we did a lot of interaction network analysis for these host protein, these HCV host proteins. And the first thing that came really clear is that HCV tend to interact what we call these hub proteins. These are protein which has a high degree of distributions, which mean that they're protein which has a direct link to many partners. And that was reported with many other studies the same. They also have significant interconnections. If you look, for example, for the host protein in general, so those which are being targeted from the HCV, they tend to be in similar biological process. Now, the most interesting thing about these studies is that we've realized that, in fact, there was an extensive enrichment for known targets of other viruses. It means that in the literature, we could find among these hundreds 80 proteins, 80 host proteins, which were also the targets of other viruses. 39 with at least 5 different viruses, and 9 of these hosts which were interacting with, or at least were reported to interact with 10 viruses. So this is really, in the last 10 years, there's kind of a concept in the field, at least those which are really working a lot on virus-host interactions, that viruses tend to interact with a very restricted number of host protein in the cells. So we've identified in the study 37 novel modulators, which we call cofactors, so they're important for HCV replications. And we also found a new mechanism of innate immune evasion system where one of the proteins, or one of the interactions between NS3 and a protein called CopinB1 was affecting the transport of a key protein STAT1. So these are all the proteins, the 100 proteins that we identified. I told you that most, but not, the majority of these proteins seems to be involved at least directly in the life cycle of the virus or in the replication of the virus, and this is through what we call genomic functioning or RNAi screen or studies that shows that they are involved. But for the majority of these proteins which are interacting with viral proteins, they didn't seem to have an effect on the replication directly. And so we indeed did a screen to assess whether these interactors would affect the interferon beta production under any viral infection. So whether they would interfere with the innate or the antiviral innate response. And surprisingly, we found 53 of these proteins that indeed are able to affect or to interfere with a virus-mediated innate response. And the most important group was a group involved in nucleotransport proteins, and those, I'm just showing you 3 of those, which are involved in the transport of proteins, either export or import. And indeed, we've opened a whole project, which I won't be talking today, but we're really looking at this class of proteins and how they affect directly the 2 key transcriptional factors, IF3 and NF-kappa B. And indeed, we found that there was a direct link. If you silence these proteins, you do directly affect the transport of those key proteins. So it gives you a flavor that the virus are able to hijack some process, in that case nucleotransport proteins, which are involved in key regulator of innate immunity, and they can maybe use it for their own replication. Sometimes they use these transporters for some other process. Okay, I'll move on to a specific example of one of the host or HCV host interactor that we've identified. Here you have a list of the proteins which we've specifically identified as core interactor. And we focus on one, which is HSD17B12. It's, I won't get into the detail of that enzyme, but roughly it's an enzyme which is involved in what we say very long chain fatty acid metabolism. So from, let's say, a C16, a palmitic acid, to greater C20, C30, these very long chains. Interestingly, in that same list, there was another enzyme, as you can see here, that was also involved in that kind of a complex. There's a certain number of enzymes which are involved in this process, as well as a transporter, which is specifically involved at the microsome in the beta oxidations of these very long fatty acids. So you see, from the list, you have an idea that this pathway seems to be hijacked by these kind of proteins. The interesting aspect, as I've mentioned to you before, is that that proteins that we've discovered was interacting with HCV was reported in the past to interact, as you can see here, with many other viruses, and not just from the flaviviridae virus, but from other group of family with flu, measles, RSV, and even HIV. So just to highlight that these proteins are often targeted by many different viruses. So this protein is indeed involved in the elongations from C16 to up to C18, and it's, as you can see, it's very complex, but the protein has a really key role in being able to generate all these fatty acids. So if you look at the virus replications, you see that when you silence or you deplete cells from this factor, we can see an increase in the level of RNA inside the cell. So if we monitor the intracellular concentrations, we systematically see quite a good increase in RNA, which we don't know at this point characterizing what type of RNA it is. But the major impact, as you can see, is the release and the infectivity, which is significantly decreased with the silencing of that proteins. Now I'll skip the rescue. We can rescue it with a protein, but the interesting aspect is that we can rescue it with oleic acid, which is the first, one of the first fatty acids being produced under this enzyme. And you can see that at the bottom, that if you deplete the cells and then you provide them with the oleic acid, which is the next step, you can rescue some partially for the infectivity, but for some of the replications. And so by blocking this enzyme, which I've mentioned to you before is a key enzyme, we can rescue it with the presence of oleic acid, which is probably responsible for many other fatty acid and lipid complex which are involved in the replication of that virus. It also affects the lipids droplet, and for those of you working on HCV knows that these lipid droplets are the sites of HCV assembly and are very important, and we believe that by depleting this enzyme, the cells need some fatty acid and will probably exhaust their lipid store to overcome this shortage of very long fatty acids, and that may contribute to the severe reductions in assembly and particles that we're seeing. It also seems to destabilize what we call these membrane web, and if you use an antibodies that recognize a double-strand RNA in infected cells, you see these very nice, on your left, I guess, these very nice dots, which are really characteristic of membrane web. And when we silence this enzyme, we see like kind of a disorganized replication factory. We have some biochemical evidence that confirm these data. So it seems to affect not only the assembly site, but also the replication site. And so this, by targeting this enzyme, which seems to be playing a key role in the elongation factor, we're able to affect the replication assembly, factory integrity, lipid droplet dynamic. We are affecting, and I didn't have time to show, the level of the core proteins. And now we're looking into flu, and it has a same effect on the nucleoproteins for flu. It affects the encapsulations, the RNA encapsulation and envelope, not only of HCV, but flu. And obviously now we're starting to look at many different members of the Flaviviridae, which we expect will be the same. So it gives you the idea of the broad, some broad kind of activity that it can be obtained through the depletions or the inhibition of that enzyme, with no significant cellular toxicity, at least by knockdown. So by knocking out with gene editing, we'll see how this enzyme is very, very important. But it gives you a flavor that it's possible to target some key proteins which may have an impact on all the member of the Flaviviridae and maybe other family. The other aspect, and we're working on this type of model where we say by acting on this particular cellular proteins, we increase the level of RNA, and indeed, even by using HCV and a competent HUH7 cells, where upon infection, normally NS3-4A is very efficient in cleaving MAV and cleaving all the antiviral pathway. By silencing this enzyme and the increase in RNA that we detect, we can see an increase in interferon beta production. So we're talking about a kind of approach could be a replication on, so increase the level of RNA that can be sensed by many different receptors in the cells to promote the innate immunity and to block the propagations of this virus. So that was just to give you a flavor. We have many of these hub proteins, which are cofactor for HCV, which are being reported to interact with many other viruses. So we believe that we have identified critical proteins that could be good targets for several virus life cycle. Sometimes these targets or these proteins, these hub proteins, which are obviously essential protein, may not be the one that should be targeted, and that's why we're trying to decipher the mechanism and their interaction subnetwork in order to identify proteins that would be more druggable from point of view of drug discovery. And we will focus mainly because we believe that for these broad activity that it's probably better to start with members of the Flaviviridae as HCV, dengue, West Nile, or Zika, which has been recently described. But clearly, probably would be extended to other RNA viruses, especially those which are being reported to act with our collections of HCV interactor. So this type of project, what we've learned from HCV as a model, I think is extremely important in terms of giving us a basic knowledge of these pathways that are hijacked by viruses, and clearly will pave the way to a novel class of broad-spectrum antivirals. And I think I probably went through. Thank you for your attention.
Maybe one or two questions for Daniel. Any questions?
For Alain?
For 17B12, are there any of the lipid-lowering drugs, for example, having an effect on the level of this protein?
Well, we screened and we got an effect of most of statins. We have another interesting story with this. So statins do indeed affect the interferon beta response, but not all of them.
Hey, very nice talk. I think one of the interesting things with hepatitis C is, as you sort of demonstrated, it took us 20, 30 years to get to the point of interferon to the first generation protease inhibitors. And then as sort of the virology groups that were working in HIV infection moved to hepatitis C in this big concerted effort towards rational-based drug design, sort of led by you and others, we had this, in 3 years, we had this massive sort of advancement in terms of therapies for hepatitis C. So I guess I'm just curious, as the virologists working in hepatitis C are now looking for other viruses, the problem is I guess there isn't that motivation in terms of the market to focus on some of the other viruses. But I'm just interested to hear your thoughts about whether a similar potential exists for dengue, Zika, other things to move that workforce to really target these other viruses that have been around.
I think it's clear that what we learn from HCV, just take the example of the NS3 protease cleaving MAV, for example, opened an entire new view of the innate sensing and signaling pathway. So I think there's a lot to be learned if we can get the proper funding. That's the issue. Because if you have an HCV project, it doesn't fly much. But there's a lot to be learned, and especially for Flaviviridae family, I think it's, we maintain a huge effort on trying to better characterize these virus-host interactions using HCV as a model, understanding what it does, and I'm sure it's going to be we will be able to apply it to West Nile, to any other Flaviviridae, as well as for any RNA viruses. Because, like I just described, I took this example because lipids are used in every RNA viruses that replicates in the cytoplasm. They need these membrane web, they have to manipulate those lipids. These lipids are also involved in signaling. So, these are key pathways that are used for many, many purposes, and that could be useful for many RNA viruses.
That's great. Thank you so much.
Short questions, short answers.
Yeah.
Good question. Okay, how are you going to make a broad-spectrum drug specifically targeting the virus-host interaction?
It's going to be mechanism-based, as I always did for the last 20 years. So I'm not going to screen like most academician does, just screen cell-based and get the compounds, which you don't know how it
I guess
works.
that's my point, though. To be that specific,
Yeah.
can you be broad at the same time?
You work for targets which has a high dependency on infected cells. Okay, they're either relocalized, there's a subgroup of proteins that may relocalize, that may be increased. So you look for a high dependency where that subgroup of proteins and complex, that's why we're gonna work on the protein interaction network of these, will allow you to define very specific to infected cells, not to all the cells.
Okay, so I'm going to follow up on Jason's question. I was going to say you did not answer the main question that we wanted you to answer.
Okay.
So quickly, apart from using HCV as a model for other viruses, is there still something that we need to study in HCV from your opinion and your expertise in the field?
I think from a drug discovery point of view, there will be probably some subgroup of patients which won't be responsive to the treatments, but it's clear that in terms of therapeutic need, I think we have to seriously consider how we can use this information to tackle some other viruses. That's sad to be saying. And after 20 years, I can tell you, after 20 years of investment of my own life, and I've been asked at BI to move on from HIV. I had worked 7 years at BI on HIV. I was asked to move on other viruses, including HCV. And we didn't have any choice at that time. We had some opinion leader people that, those are people that borrow your watch to give you the time, that tells you that you have to move away. Sorry for the long answer.